Good afternoon, everyone, and welcome to another Ageless webinar. This is one in a series of What is Aging? And it's about why we age, how we age, and how to stop it and eventually reverse it. I'm here today with Dr. Bill Andrews. Dr. Andrews has been searching for the fountain of youth for many, many years. And he's been working on the aging problem for a long time, over 20 years. He does everything from research to experiments to analysis, and has a deep understanding of what aging is and how we're going to beat it. He is the president and CEO of Sierra Sciences and has held a wide variety of positions over the course of his career. Thanks a lot for coming on here, Dr. Andrews. As you know, this is a continuation from our first webinar, which was with Dr. Ian Hale and Dr. Jose Cordero. And so today we're going to hear your theory on what aging is and what we can do to stop it and then reverse it. So thanks for, really appreciate you taking the time today as well.
Well, let me make 2 points. When I've been studying aging for a lot more than 20 years. I started my own company to focus totally on aging 20 years ago, but I was doing aging research back in the mid 1970s. So I've been on it for quite some time. I've just been obsessed with the whole subject of aging.
So almost 50 years then, or around.
Yeah, no, well, and as the story goes, it's in several different documentaries and stuff like that. I got fascinated with aging and trying to solve that problem back when I was even 10 years old, when my father put the seed in my head. My father was obsessed with that whole subject. So all through high school and college and everything, everybody used to know me as the guy that wants to cure aging.
Nice. That's a long time for sure.
The other thing, you called it a theory about why we age. I kind of think of it as best guesses. And so Jose and Ian had their best guesses, and I have my best guesses, but I'm not going to say they're wrong at all. I'm going to, I just hope one of us is right. But as I would say later on in the talk, we'll never, ever know really what aging is until somebody can create some type of non-aging. Okay, so when we have people that don't age, then we can start comparing them to the people that age, and then we can figure out what aging is. But right now, it's just a complete dark. So
yeah.
The bottom line is, we don't know what aging is. It's there's a lot of biomarkers of aging, and there's been biomarkers around for hundreds of years. I mean, all the way from grip strength to cognitive tests. But lately, there's been three different aging markers that have dominated, and those are telomere length and DNA methylation and IgG glycosylation. Those things have become very popular. People are getting those measured a lot to determine their aging. But in reality, none of those, not even telomere length, which is my specialty, none of those has actually been shown to have anything to do with aging. They've only been shown to correlate with aging. So again, it comes back to what's our best guesses. And people call DNA methylation epigenetics, but it's actually not epigenetics. Epigenetics is the study of genes being expressed, which genes are turned on and which genes are turned off, and how much they're turned on and off. Whereas DNA methylation is actually a mechanism of epigenetics. So it's an epigenetic mechanism. But it's still, epigenetics is really the biggest thing about what aging is, and really, what are the things that affect your epigenetics, turning genes on and off. And telomeres is also one of those things that do it. So
that's a good point.
So
yeah, just on that point real quick, I think you're absolutely right in the sense, I believe even after we've stopped and reversed the aging process, there's still going to be partially a mystery. And I think you see that a lot in engineering, where you get something to work, and a lot of the time the engineers don't even know exactly how it works. And we're seeing that in the field of AI today, even.
Well, that's exactly what I meant. I mean, when we actually start reversing aging, that's when the research starts to try to figure out what is aging? Okay, but we don't have any non-aging going on. Okay, so we really can't, unless we talk about some other animal like the lobster or the humpback whale or something. But but they're so different from humans, we really can't it's like comparing apples and oranges. But yeah, so when we actually reverse aging, then we can start asking the question, not just answering the question. We start asking the question what aging is. But what we do know, the one and only thing we truly can say about aging right now is that if I showed you a picture of William Shatner, young and old, everybody would know which picture was taken first. Okay,
Right.
so there's something going on there. We just can't nail it down. We don't know exactly what it is. But people like to make aging a lot more complicated than that by talking about different diseases and things like that. But even young people get those same diseases. So they're really not markers of aging. They're markers of something else. Okay, and it's a really big mystery. And so when the FDA or the National Institutes of Aging say that they won't do clinical studies to study drugs that treat aging because aging isn't measurable, I'm the first one that agrees with that. So that's why I think their focus has to be on treating aging-related diseases.
Right.
But getting back to so what is aging? The first answer is we just don't know. Okay, nobody really knows. And so, but I want to get into a little bit of other things like evolution and stuff like that, then come back to that question. And after I explain a few things, then things are going to be different. But one thing that people are always asking is, if aging is so bad for us, why haven't we evolved to not age? Okay, and that's things like, supposedly evolution is making us better and better and better. Why do we still age? Why haven't we evolved to not age? Okay, so that's really a misconception about evolution. People don't understand what evolution is. But evolution actually did the exact opposite. Evolution actually made us age. Okay, so made is a wrong word, because I always say evolution is not a force, it's a result. But as a result of evolution, we found that we developed an aging process for a purpose. And that's because it's aging, or evolution is not something that benefits the individual within a species. It's something that benefits the species in general. Okay, so whole idea of evolution is that it's some animals were one thing, some animals were another thing. The environment suddenly changed. Some animals survived that environmental change than others. So it's really the ability to survive an environmental change is what evolution is all about. And a really good example is giraffes didn't evolve long necks to eat leaves on tall trees. Okay, they already had long necks. It's just that the ones that had short necks starved to death. Okay, and so the ones with the long necks could breed, and the genes that created long necks became more dominant. But it didn't. The giraffes didn't actually force themselves to get long necks to reach the tall leaves. That was just a selection for genes that already existed. And that's a key thing about evolution. And so what I want to do is I want to kind of get into evolution, because that is, I think, really key. And I think it's where a lot of people get confused. I'm not really aware of anybody in the anti-aging field. The exception might be Michael Rose, okay, who's actually has a background in studying evolution. Okay, so evolution, there's two branches of evolution. One is the what and when. Okay, so that's, you look at an evolutionary tree, you see that this animal evolved from this animal at this particular time. A lot of people, that's all they think about when they think about evolution. Who evolved from what and when. Okay. The other branch of evolution is the how and why. how did it happen and why did it happen? And that field of evolution, that branch of evolution is called population genetics. And my PhD is in molecular and population genetics. And so population genetics is the understanding of why and how evolution occurs. Molecular genetics is how to control that evolution. So humans are the only animal on the planet so far that have evolved to the point where we can actually control our own evolution. And we haven't been just controlling our own evolution, we've been controlling everybody else's evolution too.
Yes, especially the rats.
What happened with rats?
Well, I mean, we're definitely controlling their evolution in the sense we're running experiments on them and not really allowing them to evolve naturally anymore.
Well, I was going to say especially dogs.
Dogs, that's
Okay.
true too. Yes.
Dogs were all from wolves, and they didn't evolve by nature. Humans evolved them. Now we have these small little poodle with curly hairs and things like that that are actually evolved from the wolf, and nobody would know it. But
yes, we're definitely having an effect. So let me ask you a question related to the evolution and aging. So do you think that evolution created the aging mechanism as a benefit to the species, or do you think it could potentially be more of there was no selection mechanism past the age of say 30, right? All of the selection, for a giraffe it was long neck, for humans it was mental ability to hunt and gather, maybe attractiveness, different things, those things that allow the humans to pass on their genes, but there was nothing that selected against aging really past a certain age. There was no reason to select or to create a mechanism that would say cure aging. Evolution had no reason to. It had no reason to, because as soon as someone hit 30, they've passed on their genes. They've probably already, back in the day, they've most likely already, lived most of their life, and they're now useless to the species as a whole from an evolutionary standpoint. Is that that's the way I
kind of, I'm going to come to that exact same subject, but the I would put it more at 20 years old. Okay, so
20.
so and I would also say evolution is not something that recently happened with humans. It's something that we have to go back to ancient humans and even pre-humans to really talk about the evolution of humans. But like ancient humans, we had children when we were 13, okay?
Right.
And then an ancient human was often fending for themselves by the time they were 7. So by the time you are 20, you have already raised a child who can now fend for themselves and don't need you anymore, okay? So, so at that point, I mean, you're still useful, but when you think about the survival of the species in a rapidly changing environment, that changes everything. And if that's what I want to do. So I was talking about the giraffes, and the giraffes, there were short-necked giraffes and long-necked giraffes. And then the short ones died out and the long ones survived. Okay, but what that's saying is that there was variability within the species. Okay, so there were different options. So if the environment changed, like the leaves that giraffes want to eat turned out to be higher up on the tree, at least because of the variation within the species, there was a chance that some of them would survive. If there was no variation within the species, they wouldn't survive. They'd have a less chance of surviving. And so the variability comes from genes, and the more variability that occurs in the genes in a species, the more likely it is to survive a rapidly changing environment. Now, that hasn't gotten to aging yet, but let me just summarize the history of the evolution of variability, okay? Because that's what evolution's been all about. Okay, so when life first started, everything was asexual. Okay, so their genetics was exactly the same. The only time there was ever any changes in the genetics was when a mutation occurred. Okay, and 99% of the time, a mutation would be harmful to the species. And so it would make the species sick. So it was very unlikely that a mutation would survive. Okay. Then a process evolved called ploidy. Okay, which means that these asexual organisms suddenly had 2 copies of every gene. So they like 2 chromosomes essentially. And that allowed a mutation to occur that could be lethal, but still be able to survive in that species. Okay, because it still had the other good gene that was unmutated in the same species. Now, how does that benefit the species? Well, the mutation might be bad for the present environment that they're in, but when the environment changes, maybe the mutation will survive better in that new environment than the original gene variant. And so that made it so a species was more likely to survive a rapidly changing environment by having ploidy and being able to have more than one version of each gene. The other advantage of having the mutation survive is that sometimes you can have a mutation that's bad for you, have a second mutation of that same gene that makes it actually good for you, and sometimes better than the original. And so that opportunity occurred too. So that was a step in evolution to the evolution of variability. So throughout the history, that's the main thing: increasing our variability, increasing lifespan, life forms variability, increase their chances of surviving a rapidly changing environment. The next thing that happened in evolution was sexual reproduction. Okay, as soon as sexual reproduction started, that allowed different species to shuffle their genes. Okay, not different species, but individuals within the same species to shuffle their genes. That increased variability tremendously. So every time 2 organisms breeded and they'd make a new organism, that new member of the species would have a different gene variation that ever existed before. Okay, so this was increasing variability tremendously. Now we come to the most important part, because first of all, the sexual reproduction evolved about, I want to say, 1 billion years ago. Okay, but a lot of people, I'm thinking like George Williams, I think his name was, in 1957. He published a paper in 1957 where he talked about the evolution of aging, and I think he got it right. And a lot of people need to read that study. But the reason why we aged was to make it so that the older members of the species won't compete with the younger members of the species to shuffle the genes, or to breed. And that was a big problem. So you get more gene variability by letting the offspring interbreed than by letting the older adults rebreed. Okay, so once you have shuffled your genes and passed it on to offspring, you're actually interfering with gene shuffling. Okay, because you are rebreeding and competing with the offspring that won't get a chance to interbreed. So it turned out, so if you have one species, let's say you have 2 species that are 100% identical, except one ages and the other one doesn't age, let's say by aging, some way to get rid of the old. Okay, the species that gets rid of the old was more likely to survive a rapidly changing environment than the species that didn't. Okay, and that's why we evolved. Like every species that exists right now, sometime in their evolution, evolved a way to get rid of the old. Okay, this is actually something that is well known in evolution biologists. People who have studied a lot of evolution, but tends to be forgotten or ignored, or maybe never even known by a lot of people in the aging field that tout themselves as evolutionary biologists, but they're PhDs in physics or something like that.
So let me ask you a question.
Yeah.
So I get what you're saying. So is this true today in other species, non-human, in say like some bugs or random insects? I mean, is this this concept that you're describing, I mean, if you observe a species that's non-human, like, are you going to see this phenomena in some way? I mean, has that somehow been proven? Because yeah, I guess that's my question. Like,
Nothing's been proven. Nothing.
not
It's
been
all,
proven.
it's all
Okay, so
best guess. This is all best guesses. But
okay,
it doesn't make sense. But remember, you know, evolution is always going on, and when something evolves off a branch of a tree, the original branch doesn't disappear. Okay, unless that species ended up not surviving a rapidly changing environment later on. So a lot of, so that's why most insects have programmed cell death. I don't believe that their aging process is anything like humans. I mean, all my work with fruit flies and roundworms and things like that, their death is too programmed to the date, almost to the exact date, when they're going to die. Okay, so it's like it's a built-in mechanism to get rid of them. Okay, that's to increase the shuffling of the genes. Okay, that's the only reason. And you see this especially in animals like that. The more advanced, mature animals that have been around for a while have a better chance of breeding because they learn the ropes and compete with the younger. Now, there's also the subject of what are called K strategists and R strategists. Okay, this is in the evolutionary tree. There was a time when the decision was made to either become a K strategist or an R strategist. A K strategist is a type of animal that has very few offspring, but then does a lot of work to take care of that offspring, has few offspring, takes a long time to develop that offspring, and lets them go. The R strategists are the ones that have thousands of offspring and hope that a few of them survive. And the parent, as soon as they lay the eggs, leave and never see them again. So they fend for themselves. So, it's hard to really compare insect biology to human biology just because of that major difference that I just described. But evolution is going on all at the same time. But evolution doesn't work in a serial way. It works in a parallel way where you get a branch, both forms survive.
yep.
Actually, one might become extinct, or maybe they both survive, or maybe they both go extinct.
Absolutely. No, I think it's a good point, and it's an interesting phenomena, and I think I definitely could see how that would be true. I mean, I still my guess is still that evolution, through the passing of those genes over the years, ever since you had sexual reproduction, is that they were passing along the best ones, survival of the fittest, and there's just nothing, maybe at 20 there's nothing to really pass those on more. There was just no developed mechanism, and evolution was only so good, right? So now, I believe that things, like, over the many years as an individual human, it's just things just break down. Evolution, obviously you can't even think about evolution for like a single person, right? It just doesn't make sense. So my thinking is that it just kind of, yeah, I guess I'm kind of thinking through if that mechanism is still happening to some degree, within the human species.
Well, humans now control our own evolution. Okay, I mean,
That's true.
it's like you can't break down evolution into a few thousand years. Okay, I mean you can, in some extent. For instance, poaching of elephants for elephant tusks has now made it so that a tremendously larger percentage of elephants in Africa and other places are born tuskless. Okay, because there was a selection for that gene. So we do see evolution on that scale, but it's more common to see evolution on very long-term scales. I'm trying to understand what you just said, because it's too many times you made it sound like evolution is a force and something that happens for a purpose. But it doesn't. It's a selection for, it's a result. As I say, evolution is not a force, it's a result of selection, survival of the fittest. And so it's everything about evolution is the ability to survive a rapidly changing environment. And when I say environment, I mean even humans. Okay, the worst, in fact, I think the worst evolutionary disaster that ever occurred to this planet after the asteroid that killed the dinosaurs is the introduction of humans. Humans have probably done more to destroy and make animals go extinct and things like that than anything else since that asteroid that killed off the dinosaurs.
Yeah, I definitely see. I mean, I definitely see your perspective. Like, I think of it like, say there's two planet Earths, right? Dinosaurs got killed off, humans became arose. In one planet, they don't have aging, let's just say, right? And to your point, at that point, the older species would be competing with the young, therefore you would get less variability. So in turn, that planet Earth, let's just say, there would be less gene transfer, there would be less variability amongst that species. So that human species would have a less chance of surviving, in theory, than the normal planet Earth, let's say. Is that kind of how you're thinking?
Except that we're talking about hundreds of thousands of years. So
Over the course of, millions or billions of years.
and I can tell you one thing, if there was a planet where there was no aging, I guarantee aging would evolve. Okay, because if the environment's changing on that other planet, the species that are more likely to survive are the ones that shuffle their genes the most and therefore have the most variability. And the way to do that is to ensure that the offspring have a better chance of rebreeding than the older generations, are called the forebearers. Okay, the forebearers rebreeding. So evolution has evolved in a lot of different species in a lot of different ways. And that's one of the reasons why I say it will happen on the other planets, because humans have evolved a different way of getting rid of the forebearers than other animals have. Okay, there's multiple ways of getting rid of the forebearers, but every species evolved that because the species that had the ability to get rid of the old had a better chance of surviving the rapidly changing environment. And let me make one more point. People often say that we age, well, especially in one of the theories about telomere shortening, in which I'm not planning to spend a lot of time on today, that the reason why we age or have telomere shortening is to keep us from getting cancer. And the biggest, I can't say that's not true because everything's best guesses, but the problem with that theory is that cancer is good for the species. Okay, cancer is a great way to knock off the old because it takes cancer so long to get big enough to kill off the organism that you've probably already raised your young. There's no evolutionary advantage to living longer than it takes to raise your young. And usually you've raised your young before the cancer knocks you off. Okay, so cancer, there's a lot of theories out there that just some people don't have enough experience in evolution to really understand.
Interesting. So just to clarify, so the shortening of telomeres leading to cancer was developed by evolution, you think, or was not? Just
the shortening of telomeres,
leading
does
to
cause
not get
cancer.
cancer. The shortening of telomeres as a mitigation of cancer. You're saying that was developed by evolution. You're saying that was not.
The shortening of telomeres was something that was selected for.
Selected. Okay.
Because it was a way of knocking off the old in humans, but there was never an effort to actually make it cause cancer. Okay.
Okay, I got you.
When the short telomeres get really short, you do have chromosome rearrangements that increase your risk of cancer. But a lot of people feel that keeping telomeres long is what actually causes cancer.
I've heard that before.
And so, but that's why I'm saying is that from an evolutionary point of view, that doesn't make any sense because you want the survival of the species is best if you do knock people off or animals off with cancer. Let me, because I know we're running out of time already, but
no, we're good. We're good.
I said it before. So we talked about why we age, and how we age is kind of like the next subject. And it's like there's a lot of reasons why we age, but I always ask, is there some kind of kingpin that controls the aging process? And that again, as I said at the beginning of this thought, we'll never know until we figure out a way to stop aging or reverse aging, and then be able to compare, okay? Because there's no way to know what aging is. There's no way to know what causes aging until we know what aging is, things like that. So it's all, again, best guesses. Now, one thing that I can say for certain, because besides my PhD in evolution, population genetics and molecular genetics, I also have a very strong background in statistical theory. And the big problem with the wear and tear theories on aging and the accumulation of damage or accumulation of junk theories on aging, I'm not saying they're wrong. I'm saying I actually do believe those are actually occurring on, and I think we need to get rid of those to improve our health. But to think that they might be the sole causes of aging, or maybe one or two together might be causes of aging, is got a problem in that if wear and tear and accumulation of damage and things like that were the true cause of aging, those are kind of random events, okay? And so when you have random events, you always have a wide distribution, like in the bell curve of when people die. You're going to find it never actually intersects zero, okay? And, there's 7-8 billion people on the world right now. Well, theoretically, there's been like 100 billion people that existed on this planet ever. Okay, that's a lot of people. Okay, so with that many people, you would have expected some of them to live to be 1,000 years. Okay, if wear and tear or accumulation of damage or accumulation of junk was the main causes of aging, because those are random events. And so some people would die really young of aging, some people would die really old. There'd be a few that hung out for 1,000 years. Now, the Bible and things say that there were people that lived to be 1,000 years old, but why aren't there any now? Okay, well, in the 7 to 8 billion people that exist on the world, I think it's 7.7 right now that exist in the world. None of those are living to be past like 120 at the most. None of them actually reach 120. So it says to me that there's actually some mechanism, just like in insects. There's something that actually, a program that says they have to die at a certain time. And in humans, it's not the same as insects, but in humans, I believe, this is now, and let's say, I don't like using the word I believe. I like to say, I wouldn't be surprised if we find that the thing that knocks off the yolk is telomere shortening in humans. And that's not a wear and tear. A lot of people think of it as wear and tear because they keep talking about telomere shortening or degrading or eroding or things like that. Whereas, probably less than 1% of telomere shortening has anything to do with that. And the fact is telomeres don't actually shorten at all, okay? They get shorter because when new DNA is made, when a cell divides, the new DNA is made shorter. It's not degraded or shortened. So it's called the end replication problem.
Mm-hmm.
Now, what's nice about that actually makes it an actual ticking clock, okay? That is a mechanism that is very well understood. So there's a ticking clock that's showing the aging of humans that has no wear and tear involved at all. I mean, everything wear and tear. So telomeres can be shortened by wear and tear, but it's probably accounts for like 1% of all of the telomere shortening. So, so that's why I focus on telomeres. Now, it's still a best guess, but what I'm trying to do is I'm trying to create the ability to ask the question and then answer the question. I want to ask the question, does telomere shortening have anything to do with aging? And there's a lot of things about it. I mean, about telomeres do limit. See, this is, as I was saying, how wear and tear can't limit our lifespan to a narrow range. It actually, wear and tear will cause some people to live to be 1,000. Whereas this telomere shortening, actually, mathematically, you can calculate that a human can't possibly live beyond 125 years, okay? And that and then because of the fact that there is accelerated telomere shortening from the wear and tear kind of processes, nobody actually lives that long. But it's like even if you had the perfect genetics and led the healthiest lifestyles, you can't live beyond 125 years. Okay, that's just impossible. And I'll tell you, I don't want to spend a lot of time on telomeres, but there is, if people go to my website, I do have a video. It's a 2-hour video, but it's really good because it's got a table of contents in the middle at the beginning, so you don't have to listen to the whole thing. But it goes through everything. It's called Everything About Telomeres, and you can hear all about my best guesses. And I think I even call it my best guesses in that video.
Yeah, that makes sense. I mean, so okay, when I think about this damage, these are really downstream results, right, or effects of some upstream process. I think of like some process like lipofuscin accumulation, something that's, you know, accumulating, say, in the cell, some junk, as sort of this result. Now circling to like the telomeres, you know, I've heard it's all, well, one thing I've heard is about the rate of telomere decline versus the actual length. You know, maybe let's start kind of with that question. I mean, do you think it's purely based on the length, or do you think it's based on the rate? I mean, they kind of go hand in hand, but what are your thoughts on that?
It is both. I mean, that paper, there's only one paper that's come out so far that said it's the rate of telomere length, not the length of telomeres. But it's the rate of telomere shortening is going to increase your it's going to make you die or age sooner.
Length, yeah.
But it really still comes down to the length. And there's extremely good theories as to why the length of the telomere affects your epigenetics. Okay. I mean, anybody who's taken genetics 101 knows all about enhancer sequences and how enhancer sequences can be 10,000, 100,000 bases away from a gene, actually regulated gene, by the DNA folding over and coming in contact. Well, the telomere can do the same thing. The telomere can be thought of as an enhancer sequence and can fold over and, like a magic wand, interact with genes, turning them on and off. But the shorter that telomere gets, the less far it can reach. And so that's playing a big role. And why do mice? Well, see, mice is not a good example, because contrary to popular belief, mice do not actually age by telomere shortening. They have telomerase on in all their cells, and their telomeres are so long you can't measure them. Even the ones with the shortest telomeres, Mus spreadus, has telomeres the length of humans. So why don't they live to be as long as humans? But it's telomeres actually, I believe, have very little to do with aging in humans. But telomeres might have, like, moonlighting activities and things like that maybe increasing the telomerase activity in mice might have an effect on their lifespan. But I don't think it has anything to do with their actual telomeres.
I see. So remind me, so we have a telomerase. That's the enzyme that can repair the telomeres.
Yes.
The caps of the chromosome. Okay, so
wait, no, stop.
Oh no, I may have
use the word repair. Do not use
not repair. They don't
There's
repair.
no damage.
Okay.
Dysfunctional telomeres and all these terms. These are vague terms that people use to explain it all, but it's really not. Because it's complicated, but it's really the end replication problem. It's not a repair. Repair is a very minor part of the effect of telomere shortening.
What is the telomerase doing to the telomeres?
It's lengthening the telomeres.
Lengthening. Okay. So let's just say hypothetically you had telomerase, super telomerase, let's call it, and that super telomerase was in every single cell in your body, from your heart to your, well, I guess brain, to your brain. Let's just say hypothetically, this super telomerase could maintain the lengthening process of your telomeres so they don't get shorter. At that point, like, what do you think would happen to the cells? What do you think would happen to that organism? What would be your guess?
They would live healthier longer, and the human would live healthier longer. But here's another caveat about all the aging process. There's other causes of aging besides telomere shortening, if telomere shortening is one of the causes of aging. I think of telomere shortening as multiple sticks of dynamite that are burning inside your cells, and there's oxidative stress, there's mitochondrial dysfunction, there's accumulation of junk, there's all kinds of different things going on that cause aging. But in humans, the stick of dynamite with the shortest fuse is telomere shortening.
And do you think that if you were, say that species lived, you have the, let's just say they were, the same person, hypothetically again, two people,
identical twins,
two people, they both, one of them had the super telomerase, the other one didn't. 10 years out, do you think that specifically their epigenetic age would be significantly different that 10-year out?
I wouldn't be surprised if that was true.
Okay.
I'm not going to say I think.
That's what I meant. That's what I meant. Would you be
still
surprised?
all theoretical? Yes, but
Okay.
that's the model that I'm trying to test right now. So I need to come up with, and I first have to come up with a hypothesis. And in order to come up with a hypothesis, I have to have a way of testing it.
Gotcha.
And until I have a super telomerase, I have no way of testing it. Okay.
No,
so
it's a tough problem. It's a tough problem. So here's another one. So say, comparing those two methods, I mean, obviously we don't have a super telomerase yet, but thinking about like the
those pairs would disagree.
Oh, maybe we do. Oh, maybe we're close.
I agree, but they're still in studies and they're super expensive and things like that. But hopefully we'll get some proof of concept from that.
So they're making something in the lab, basically.
Well, the gene therapy.
The gene therapy. Okay.
Telomerase gene therapy is essentially producing the super telomerase.
Okay, and that gene therapy, and I'm sure I'll ask other people, but that would be like lengthening, that would be optimizing the telomerase, that would be a super telomerase.
Yeah, that would be lengthening telomeres.
Okay.
When we do it in vitro in the test tubes, that's exactly what we see. When we infect the cells with the gene therapy, we actually see the telomeres get longer. Okay.
So maybe if I was talking to George Church just this week, well, not just myself, but a bunch, on a call, and he has a combination gene therapy, 4 of them going in at once. And, he's saying that really he had 3 areas to work on, the delivery mechanism being one of them, just like, can we get this into say 90% of the cells? So that was something interesting. But, circling back real quick.
Big problem. That's a really major problem.
Big problem.
That's the biggest problem in gene therapy right now, is getting it to a significant number of the cells.
That is true. Yeah. I mean, with lipid nanoparticles, I think that the mechanisms will improve, the delivery mechanisms. But yeah. So I'm thinking like, okay.
We're working on it. And we're using nanoparticles and lipoparticles and therapies, everything here, trying to find some way to do it. But so far the gene therapies are getting to a higher percentage of the cells than anything else, unless you use green fluorescent protein, which always gives you very different information than any other gene. I don't know why green fluorescent protein is always giving us wrong answers, but go ahead.
Green fluorescent. Okay, I'll have to look that up later. So, so I'm just comparing like the cellular partial cell reprogramming, sort of the stuff like that David Sinclair talks about in relation to like the Yamanaka factors. You got these 4 factors, these genes, they're saying, you can reset the cell. So what I want to know is, when they reset that cell, what happens to the rate of the telomere decline? I mean, would they say, oh, you've fixed this telomere problem at that point? Or
when you use the Yamanaka factors to reprogram cells, okay, and we've been doing that, God, ever since the Yamanaka factors came out. We published a paper, I think, in 2010 with Mike West on that kind of stuff. But the only thing that occurs inside reprogrammed cells that actually essentially rejuvenate the cells is the lengthening of telomeres. Okay, and it's when you use all 4 Yamanaka factors, or some versions of it have more than 4, but when you use all of them together, one of the things that happens is you induce telomerase expression and lengthen telomeres. And that's what we published about in, I think, 2010. And so that's the thing that rejuvenates. Everything else about the Yamanaka factors de-differentiates. Okay, and that's my concern. I'm very interested in seeing this research get pursued, but I think the only thing, I think one of the biggest problems with, that's why they're doing partial rejuvenation, because they do know that the Yamanaka factors are causing de-differentiation. You don't want to do something like turn a butterfly into a caterpillar, or a frog into a tadpole. That's essentially what de-differentiation is, because if you didn't at the same time induce telomerase expression to lengthen telomeres, those tadpoles and caterpillars would not be younger than the butterflies or the frogs. Okay, and so the only thing that makes them younger is the fact that telomerase lengthens telomeres. So I've been studying the Yamanaka factors and induced pluripotency and things like that since its infancy. But really, all that's doing is providing us some clues about telomere length. So really, we don't want to turn our cells into dedifferentiated cells. We just want to make them younger. Okay, and so what we have to, and I don't think partial reprogramming is going to work. I think partial reprogramming is going to cause too much dedifferentiation problems. So what we have to do is we have to figure out a way to just turn on the telomerase gene. So back in, I think 2008, I think it was, we spoke at a conference. My whole company went and gave presentations at a conference in San Francisco, where we had done a bunch of studies of taking all the Yamanaka factors, not just the 4, but the other. There's been other versions. Some have MIC in them and things like that, where we tried using like 1 or 2 or 3 in all different combinations, or 4 or 5. There's sometimes where we had 6 different factors, and we try just all combinations. So 1 or 2 or 3 or 4, all different, everything in combination imaginable. But every single time, we had some great posters on the data. Every single time, the only time we got telomerase to be produced was when we used all of them. Okay, and so there was no, we were looking for a way to separate the dedifferentiation from the rejuvenation, and we could not do that. Okay, so it's something about the dedifferentiation process that is actually causing the telomerase to go on, and we can't seem to separate the two. And so we published on that, and we did the poster sessions, and so we kind of abandoned that approach, I think in 2010 or something like that. Now to see it resurrected, and Basil's actually spending tremendous amounts of money on it, I think they're, I hope in all the best of luck, because really the #1 most important goal for me is to reverse my own aging, you know, and I don't need to be the one that's right. I've always got to make best guesses, and I'm not certain they're right. I'm worried they're wrong, and that's why I want to continue to pursue my research to try to turn on just the telomerase gene to lengthen telomeres without causing the other dedifferentiation problems that occur with genetic reprogramming.
And if you were gonna do that, I mean, would you pass that to every single cell? I mean, or 90% of the cells, if you could, like if you had that mechanism, or would you do more of an isolation, like, okay, let me do my arm, or let me do
Well,
my...
no, I would do all of the above, but we
Okay.
have research right now going on that where we are doing what do you call it, directed evolution, to try to create new gene therapy vectors that will get into all the cells. The same problem is occurring with the liposome approaches and every type of nanoparticle approach. They can't get into all the cells very efficiently. So I actually believe the best way, in fact, I think before anybody ever figures out how to get gene therapy or nanoparticles or liposomes or something like that to get mRNA or DNA to all the cells, we will have identified a small molecule drug that will get inside, because once you get a small molecule drug in your blood, it gets to every cell in the body. And we can put that in high doses enough to get every cell, this drug into every cell, and the drug would then induce telomerase expression. Because that's been the really key focus of our research forever, even though we've been doing everything else for proof of concept and stuff. We really think the small molecule drug is the best way. And we did have a lot of success doing the small molecule approach. In our best success, we found over 900 different chemicals that would induce telomerase expression in every cell in a petri dish. But the most potent one we got was one that would cause the gene to turn on about 16% as much as we actually think we need to lengthen the telomeres. So decrease the rate of telomere shortening by producing a little bit of telomerase, but not enough to actually lengthen the telomeres. So we had to stop that research after we discovered a drug called C0314818, because that was the 314,818th drug we tested. That was our most potent. We tested about 500, 600,000, but we never got anything better than 314818. But we were doing medicinal chemistry to develop even better versions of that one when the funding all dried up after the global financial crises that have occurred in the last few years.
Oh yeah, no, it happens. So let me circle back to the concept.
But that's what we're doing. That's our goal. We're trying to get that. We're trying to find a small molecule drug in parallel with trying to get gene therapies and things to work.
Yeah, I think they're both going to be promising. I mean, I think small molecules. Yeah, I think the delivery mechanisms are going to just get better and better. So I think it'll be fine. Circling back to this, and I'm sure you get this a lot, the question about cancer. So I know you were saying from an evolutionary standpoint, it wouldn't make sense if you lengthen the telomeres that would lead to cancer. But the thing that I'm not understanding, so like if my biology decreases the length of the telomere as a response to potentially that cell becoming cancer, is that a tagging mechanism? Like when that telomere starts getting shorter, does the body say, oh, apoptosis, it's coming? You get what I'm saying? Or is that like, well, is that a tagging mechanism for the body, or
actually there's a few things that you said that actually didn't quite make sense to me, but
okay.
Let me just say that when telomeres get short, it induces senescence. Senescence is not apoptosis. Okay.
Okay.
So you can have a senescent cell around for a long time before apoptosis would occur, but apoptosis will occur for some totally different reason because of the telomere. Okay. So
not because of the telomere length. Okay. Okay.
No, I get you. Maybe it's not even apoptosis. Maybe the cell just gives up or die. I don't, it's hard to say, but.
So here's a follow-up on that. So, I mean, do you think that the telomere length is sort of the core trigger to senescence, or do you think that it's sort of like a bunch of things happening at once that causes the cell?
Well, a lot of things cause senescence, okay, but replicative senescence, okay, the thing that Leonard Hayflick discovered, okay, back in 1961, where after a cell divides 50 times, it can't divide anymore because the telomere, he didn't know why. He just knew that it stopped. Okay, so from 1961 to about 1990 something, nobody knew why human cells could only divide a certain number of times. They used to say senescence is irreversible. Okay, and so nothing has ever worked. Replicative senescence is irreversible, and actually any true senescence by definition is reversible. Otherwise it'd be called quiescence. Okay, quiescence is the same as senescence, but it is reversible. Okay, so now replicative senescence was irreversible until we at Geron Corporation, after we discovered telomerase, put telomerase into cells and showed that they never senesced. Okay, they never achieved replicative senescence. And even Woody Wright and Jerry Shea, who worked at University of Texas Southwestern, they were able to create engineered cells where they could turn telomerase on and off, and they would let cells get into senescence, which are supposedly irreversible. They would turn on telomerase, and these cells would come back.
Oh, okay, they would reverse them. Oh,
so
wow.
I think there's no question. I don't think this is even a theory anymore. I think it's an absolute fact that telomeres are the only cause of replicative senescence, and that lengthening telomeres completely abolishes replicative senescence.
I can see that. I can see that.
Mice don't have replicative senescence. They have a different type of senescence that's caused by oxidative stress and other things, because antioxidants will reverse their senescence, whereas it won't reverse human senescence. And
is there another type for humans besides replicative senescence? Or?
Yeah, there's anything. Well, see, there's a whole bunch of things that can cause senescence, like damage to DNA, things like that. So once you do anything to a cell that makes it impossible for it to replicate anymore, that's called senescence. Okay, as long as it's still alive. Okay, so there's a lot of different ways to do that. A lot of them are mutations, things like that. But lengthening telomeres won't reverse those senescence. It only reverse replicative senescence, which is the #1 cause of senescence in humans. Okay, it's replicative senescence.
And do those senescent cells from replicative senescence, do those become, or I guess can those cannot become cancer cells, right? I mean, without lengthening the telomeres
Well,
to revert
they'd
back.
require additional mutations. Okay, so those cells that have lost the ability to divide, remember, lengthening telomeres does not cause a cell to divide unless it is suffering from replicative senescence. Okay, if it's senescence from something else, lengthening telomeres won't have any effect. Nothing will have an effect.
Okay.
Except for possibly other mutations that might occur that would suddenly make it so that that cell can now suddenly start dividing and turn into a cancer cell.
It's interesting. Yeah, it's I remember I heard about this concept, and really it comes down to three mechanisms. So you can either take a cell and you can completely avoid going the senescent route to begin with, or you can have it go to senescence and then flip it back, kind of the way I think you're describing, or you can have something destroy that cell. So there's 3 ways to sort of like get rid of these senescent cells that are doing damage, and the method you're proposing is kind of the second one, where it goes to senescent and then you flip it back by lengthening the telomeres.
Well, let's talk about senolytics for a second, okay?
So the third mechanism,
third method. So what happens when you destroy a senescent cell?
Well,
I don't know if you can answer it, but I can answer it.
Yeah, I mean, maybe I'll have you answer. I mean, I could answer it, but I think let's have you answer that one.
When you destroy the senescent cell, another cell has to divide to replace that cell. Okay?
Right.
And that causes telomere shortening. So as soon as the cell divides, the telomere gets short. So getting rid of senescent cells has been really beneficial in rodents and mice and stuff like that, where they really aren't suffering from replicative senescence. They're suffering from some other kind of senescence. When you get rid of senescent cells, this is my biggest worry. So a 95-year-old person, 95% of their cells might be senescent, replicative senescence.
Wow.
Okay? And so you kill off those senescent cells, there's no cells to divide to replace them. And
there's hardly any cells left.
Yeah. So senolytics is making some big waves in younger people, okay? Because it does make you look younger and stuff like that. But just like microdermabrasion, which is where you treat your face with acid or chemicals and stuff like that, it kills old dying cells. It causes younger cells to divide to replace those cells. And whenever you have fresh cell division, it always makes you look younger. But in fact, they're older. Okay, they're older if telomere length is the cause of aging. Okay, let me make that real quick. But so the telomeres are short. They're definitely, the telomeres are shorter, and so therefore that puts a limit on the lifespan. Okay, because let's say there's 2 things. Does telomere shortening cause aging, or does telomere shortening limit your lifespan? Okay, whether telomeres cause aging, that's a theory. Does telomere shortening limit your lifespan? That's not a theory. That's a fact. Okay, that's every single person that works with human cells in petri dishes in the lab know that telomere shortening makes it so that their cells quit dividing after a while, and they have to get fresh cells to continue their experiment. Okay, so and the only way to prevent that is lengthening telomeres. So in humans, there's major differences between humans and mice, and I could spend an hour on those, but. I'm really afraid that senolytics is actually going to be accelerating. Well, it's definitely going to accelerate replicative senescence. Will that be accelerating aging?
I see. Do you take any senolytics yourself?
Yes.
Oh, okay.
I mean, you only live once, right? Let's do everything. I want to be as young and healthy as possible. I'm a big believer that if I'm accelerating my aging, I will have that problem solved in at least 10 years from now before I need it. And so I highly recommend everybody do microdermabrasion because it makes you look better. do human growth hormone, which also accelerates aging by making cells divide because it makes you look better. Immune boosters accelerate aging by making immune cells divide, makes you healthy, feel healthier. I do all these things, okay, because I have a faith in myself, and not me, maybe somebody like Liz Parrish or somebody else or Mike Fossil, that are going to actually come up with some way to lengthen our telomeres so that after we've done all the damage to ourselves from all these great things we can do, we'll be able to fix the problems. Okay, so
yeah.
I definitely I want to feel healthy and happy as much as I can. So
Yeah, I have questions on human growth hormone, but maybe I'll ask those later.
let me just say one thing. I
Okay,
always
go
say,
ahead.
what's the point of living a long time if you're not living?
That is true. That
So
is true.
take your hormone, take your senolytics, do all that stuff.
Drink your beer and some tequilas.
Yeah, and have fun. Yeah, have fun with
Right.
life. But alcohol definitely causes accelerated aging, especially if you're like your liver, because it kills liver cells. And when you're young, that's okay because you always have the other cells to divide to replace them. But after the telomeres get short, those cells can't divide anymore to replace them, and you get liver cirrhosis. So it's just be careful with the drinking.
Yeah. No, it's a good point. It's a good point. So thinking about what you would really want, then you'd kind of get the cell either when it's still healthy or maybe when it's I've always have
quiescence.
when it's within the quiescent state, you would then flip it back, either flip it back or get it even when it's healthy. If you could detect like, okay, these certain cells really need more telomerase, these ones not as much, they all get like a little bit. I'm assuming like some cells would need it more. There's also so many different types of cells. But in theory, though, you would want sort of like a maintenance system to optimize the system integrity of the cell to maintain the telomeres in a better way than our cells currently do. That's kind of the main thing.
Yeah, but let me qualify one thing. Quiescence is not caused by telomere shortening.
Okay.
Only replicative senescence is.
Okay. Okay. So, but doesn't it have to go through that state to become a senescent cell, or?
No, quiescence can occur early, even when telomeres are long.
Okay.
It's other factors. In fact, sometimes quiescence is a normal process, okay, that can be reversed. Now, when you have something terminally differentiated, like a nerve cell, that's not reversible. So that's not called quiescence. But quiescence is where a cell totally stops dividing. I would say like a naive immune cell could be thought of one, except that naive immune cells are actually dividing all the time. They're one of the things that are called consistent replicators. But there are cells in our body that do go into the quiescent stage where they just sit back and just do nothing, waiting for something to induce them to go. Okay, those are called intermittent replicators, and then like nerve cells and heart cells are called non-replicators.
And do they sometimes flip back? They go into that state, and
yes, the quiescent cell can be turned back into a regular cell.
Interesting. Okay. Okay. Well, it is interesting, and I mean, I think it sounds like, we really have to solve this problem. At least, I mean, when I look at the broad 9 hallmarks and now really the 14 plus maybe some others for the ones that came out, I mean, telomeres is on there, and I think that I think that maybe it plays a bigger role. Maybe then it's not a single sliver. Maybe that is really something. But I feel like too, like, yeah, I'm sure the hallmarks are a little bit interrelated. I think about like the microbiome, which I can't say is directly related to telomeres, maybe at all, but I know it has to be important since we have so many of these small organisms on our body and within our body. I've heard even more than the cells in our body. I mean, do you think that would play a much smaller role than telomere length in terms of?
Not at all. I'm a very big believer in the microbiome. Biome is something we need to keep as healthy as possible, but we have control over that more than we do on our telomere length. Our replicative senescence, our end replication problem with telomere, we have no control over that at all. That's going to have, so no matter what you do to stay healthy, and I encourage everybody to do everything they can to stay healthy as long as possible, you're still doomed because there's nothing you can do about your in replication problem, your telomeres. That is going to cause, no matter what you do, you're never going to live to be 125 until me or Liz Parrish or Maria Blasco or Mike Fossil or somebody else comes up with a way to actually get telomerase induced in all of our cells, produced or induced in all of our cells to lengthen our telomeres. But I do believe in the meantime, do everything you can. In the microbiome, I'm actually working very close with a company. I think I can pronounce it. It's Propion right now. That's very, very interested in dealing with the microbiome problems, or at least some of the people in that company are very big names in the microbiome problem. And I am a big believer in doing everything you can to protect your microbiome.
Totally makes sense. Yeah, it was well said. And I get what you're saying. It's sort of like the telomeres give you that end date, like almost, if you can't change that, you got that end date almost no matter what. And these other pieces of the puzzle are going to either accelerate you to that end date and make it short, make that end date come quicker, or maybe push it out a little longer. But you're never going to get to 200 years old without solving this particular cause of aging.
What I always say, no matter what else you do, and I encourage everybody to do everything they can, but no matter what else you could do, aging will never be cured unless you also solve the telomere shortening problem, because that definitely is a ticking clock. I'm not going to say it causes aging because that's a theory, but it definitely limits our lifespan to a maximum of 125 years, and nobody lives that long because everybody has poor genetics or poor lifestyle habits that actually accelerate that telomere shortening.
Gotcha. Like when David Sinclair is talking about like resetting the cell to a youthful state, I'm under the assumption that he is including this telomere concept in that reset, right? You wouldn't have reset the cell without resetting.
Genetic reprogramming is lengthening telomeres. Yeah.
Okay, so he would.
David and I have talked a lot about telomeres and telomeres. I'm 100% convinced that David's a big believer in the whole field of telomere biology.
Okay. So he includes that.
But there's a lot more. I mean, right until in fact, there'll probably be still a lot of things you have to do after we do solve the telomere shortening problem. You have to still maintain that's what I was saying. There's multiple sticks of dynamite. The one with the shortest fuse is telomeres. Well, even when we solve that telomere shortening problem, we have these other sticks of dynamite that can knock us off.
Mitochondria messing up, all kinds of things.
Yeah. Hey, I don't know if we're winding up, but I said at the very beginning that I was going to come back to after I talked about evolution, I was going to come back to what is aging.
Let's do it.
And just really quickly. So now that I've explained everything, I think aging is just humans' way of getting rid of our forebears. Okay, getting rid of our forebears to maximize the gene shuffling to allow us to survive a rapidly changing environment. That's what it's for. Now, but this manifests itself as system failures of a lot of biological systems. Okay, and one of the things that a lot of people think is that the failure of these biological systems is the cause of aging. I believe that that aging is the cause of these failure of the biological systems. Okay, and so we can attack the biological systems, and I think I encourage that. There's a lot of research is going on, but we still have to attack the aging problem, and what causes aging has to really be identified. What is aging has to be identified, and that's where I think we are. But the key is aging is a failure of a lot of biological systems all at the same time.
Totally makes sense. Yeah, I'd have to agree. It's definitely a multi-pronged system that begins to fail because of many different processes. But I have no doubt that we'll be able to solve each one. We will solve each one. It's just a question of when, not if.
I just hope we figure out the cause of aging first, because if aging is the cause of these system failures, solving the aging problem should solve those too, until the other sticks of dynamite, the other fuses on the other sticks of dynamite get short.
Yeah, I mean, but to your other point, we may not know the cause until we've already reversed it. And we may still not know the entire cause either.
And no, actually, we're not going to know the cause until long after we reversed it.
Okay, yeah, that's the
That's
crazy part.
everything that I'm doing, and Aubrey de Grey's doing, and Greg Fay's doing, and all other people on doing research on aging. we all want to be able to test our theories or best guesses. Okay. And then if one of them works, then we know that works. Then we got to figure out why. Okay. And how
Yeah.
it did it. Yeah.
And it's good if it works, even if we don't know why. We were
Right.
getting it to work. That's a good step.
Maybe nobody will care why it works afterwards. Yeah.
People will want to know.
I will for
Oh
sure.
yeah. Oh yeah. Awesome. Well, really appreciate it, Dr. Andrews. Any final remarks for the audience? Anything before we're looking? We're yeah.
Oh, well, I'll tell you, my obsession with aging is personal. Okay. I want to cure my own aging. I love living. I want to live healthy as long as possible. I want to also focus on its quality is the most important thing. So, but I want to extend the quality as long as possible. Um, and my biggest interest in living longer, besides loving living, is that I think the future has so many great things to offer. And like I want to be there when we make first contact. I want to be there when we figure out, is the universe expanding? I want to figure out all these different things that are going on. It's like there's so many great things in the future. I don't want to miss out on it. And so that's why I'm working around the clock trying to figure out a way to cure aging.
Absolutely. I definitely agree. It's one of those things that, a lot of people, they try to come up with some reasoning why people should die and that aging is a good thing. But, we, and I speak for ageless partners, have never thought that way. We always think that it's a tragic, basically something that is not great. When you go into a nursing home, retirement home, especially at the top level where you have people who are getting ready to go, they're not having a good time. They're not on the beach. They're not having fun. And I think that the fact that we know that that is reversible, that tells me that we have, as a species, as a civilization, a moral obligation to pursue exactly what you're describing more aggressively.
Well, as I said, aging is terrible for the individual. You're right. But it's good for the species, survival of the species. Okay, so we are in
Trade off.
control of our own evolution now. The hell with the species. I'm only interested in the individual. I want to cure my aging. And so I believe aging is a terrible thing and I want to figure it out. But I think even though it might make it less likely that the species will survive a rapidly changing environment in the future, I think humans have evolved past that and we'll figure out solutions.
Yeah, I think with the advent of these technologies and AI, that concept will be slightly different because if you have someone who has reverse aged back, they're going to be producing a lot for, assuming that they're still valuable for the economy, for our civilization. They'll be doing a lot of good stuff. So,
I'm just worried about me. Everybody else can. Oh, no, I'm worried about more than just me. But I don't. I would never want to see people die to save the economy. Okay, and that's like I don't know. I think of it like the reason we draft kids to wars and let them get killed and things like that is for economic reasons and survival of the business. Like the country's a business, and to make the business successful, we have to go off and kill some kids. I think that's something I'd love to see done away with. And just like aging, I don't want to see people die of aging for the same reason.
definitely agree 100%. I think what our society doesn't yet realize is the economic benefit of solving aging is actually very big. Even if you can slow it down just a little bit, you're going to have less healthcare costs. You're going to have more people working for longer. You're going to be saving about 38 trillion a year, according to Andrew J. Scott in one of his papers. And I think that the U.S. government is going to at some point they're going to realize this, and that's when they're going to deploy a lot of capital and they're going to make a big difference.
Keeping people healthy is what's going to
People healthy, yes.
make it benefit the country more than just aging. Okay,
Yeah,
so
keeping people healthy and strong and able to continue to work and not have to just get old and then be more of more of a taker, just kind of taking healthcare and needing things, needing caregivers versus a producer.
Well, let me ask one more question. As I addressed this in my first book.
Mhm.
There is going to be a problem if all of us stop aging and have declining health, and stop having declining health. And that's, how will the young ever get jobs if the old never retire? Okay, that's going to cause a tremendous impact, a negative impact on the country and the world. And somebody's going to have to figure out that problem, and I'm going to let somebody else do it.
Yeah, I think the AIs are going to be doing most of the work anyway. So we're
When
yeah.
you say AIs, are you talking about robots? Because AI is a very misused term.
It's
AI
a very broad thing, but I think that if we're talking about the long term, 50, 100 years from now, I think that, I mean, unless we've enhanced our brains through Neuralink, I think that the AIs will be doing the labor.
how, because AI,
I know I'm using it broad. I'm saying like,
very
say,
misnomer. It's a lot of people are misusing the word AI, and I just want to figure out, when you say AI, what are you referring to? The robots or the computers?
it'd be a combination. So any task, say I'm developing software, or I'm flipping a burger, or I'm driving a taxi, or I'm collecting something, or I'm selling something, the AIs are going to be able to do it much faster, much better than current unenhanced humans.
The definition of AI has always been just computers. So
AI and robotics.
robots are not AIs. Robots are robots that have computer programming called AI.
AI and the
Oh,
hardware
it's
behind the
self-taught,
AIs, robots as well. That's
no,
what
it's just semantics. It's just whatever. Okay,
No,
so good.
sure. Anyway, yeah, so I think we've determined what is aging, how we age, and how to stop it. So thanks again. Really appreciate your perspective on the topic. And yeah, thanks again, and
Well, thank you very much.
thank
All
you.
right.
I'll take us offline. Live long and age less.
Yeah. Are we still going to talk, or are we hanging up now?
We'll still talk.